Welcome back. In our first lecture, we talked about brain aging and its effects on our mental abilities. And we pointed out that the brain is subject to age-related decline, just like other parts of the body. And that you can see these effects most clearly when you look at changes in processing speed, in executive function, and in episodic memory. Well, in this lecture, I want to begin addressing the question of why our brains age. I mean, what is going on in the individual brain cells that causes our brains to work a little less efficiently as we get older? It turns out that many of the changes that are happening in brain cells are also happening in every other cell in our body as we age. And in fact, there's now substantial evidence that many of these changes are actually genetically programmed. So today, I want to talk about the genetics of aging. And I want to begin with the story of the oldest human being, a French woman named Jeanne Calment. According to French census records, Madame Calment was born in 1875 in the city of Arles. This means she was born before Alexander Graham Bell made his famous first phone call, before the Brooklyn Bridge was finished, before the Statue of Liberty arrived in New York, before Custer made his last stand, and before Edison invented the light bulb. But she also lived to see the cloning of Dolly the sheep, the Oklahoma City bombing, the O.J. Simpson trial, and the founding of eBay. In fact, she lived until August 4th, 1997. So at the time of her death, she was 122 years old. Now, of course, most people don't live anywhere near as long as Madame Calment. In fact, the average life expectancy in the US in 2012 was about 79 years old. But that's still actually quite long compared to most other animals. For example, horses typically live 20 to 30 years. Cats and dogs average about 10 to 15 years. And houseflies only live a few weeks. On the other hand, some animals live a lot longer than humans. Tortoises have been known to live over 175 years. There are deep-sea sponges in the oceans around Antarctica that are estimated to be over 10,000 years old. And many biologists believe that the hydra, a small freshwater animal that looks like a tube with tentacles at one end, as well as some types of lobsters and fish, don't age at all and could potentially live forever if they didn't die from causes unrelated to age. So obviously, there's enormous diversity in how different species age. But there's also substantial consistency in lifespan within each species. And together, these facts suggest that aging might actually be part of a species' genetic program. And that brings us to three questions that I want to address today. First, is it really true that some aspects of aging are genetic? I mean, is there any solid scientific evidence to support that claim? And it turns out there is, and we'll spend some time talking about it. The second question is this: why is aging influenced by genetics? I mean, why hasn't natural selection gotten rid of the genes that make us grow old and die? Shouldn't our genes try to make us live forever? So we'll also discuss scientific theories about why our genes influence aging. And third, how does this work? That is, what are some of the genetic mechanisms that lead to age-related deterioration? There's a lot we still don't know, but scientists have made a number of new discoveries in the last few decades, and we'll review some of the most important. Let's begin by talking about the first question. Are some aspects of aging genetically programmed? A number of scientific studies suggest that the answer really is yes, and one piece of evidence comes from the study of twins. Identical twins are so called because their genetic makeup is virtually identical. That's why they look so much alike, and why they share so many traits. I mean, I actually have an identical twin myself, and so I can attest to the similarities. Not only in physical traits, but even in mental and personality traits. Fraternal twins, on the other hand, are no more similar genetically than any other siblings. So they tend to share a family resemblance, but they don't look identical. And the same is true for any trait that's influenced by genetics. If a trait varies from person to person and is significantly influenced by genetics, then that trait should be more similar in identical twins than it is in fraternal twins. In particular, if our genes really do influence our lifespan, then we would expect the lifespans of identical twins to be more similar than the lifespans of fraternal twins. It turns out they are. Dr. James Vopel and some of his colleagues in Denmark and Minnesota analyzed the lifespans of 2,872 Danish twin pairs who were born between the years 1870 and 1900. And sure enough, they found that the correlation between age at death was higher in the identical twins than it was in the fraternal twins. And based on their data, they estimated that about 25% of the variability in lifespan could be attributed to genetic factors. So apparently our genes really do influence how long we live. But scientists have gone much further than that. By now, they've actually identified dozens of specific genes that influence aging in different animal species. One of the first studies was published in 1988 by Thomas Johnson and David Friedman at the University of California at Irvine. They studied lifespan in a small roundworm called C. elegans. What they did was to separate worms that lived a long time from worms that didn't. And by selectively mating those long-lived strains, they were able to show that a single genetic variant was associated with a roughly 50% increase in average lifespan, and that it almost doubled maximal lifespan. They named the underlying gene age1 because it was the first gene to be associated with significantly longer lifespan. Now, when this discovery was first reported, many scientists assumed that the age1 gene was not actually regulating aging itself. Rather, they thought that it was making the animals less fertile, and that the animals were compensating for their reduced fertility by living longer in order to increase their chances of reproduction. And at the time, even the authors themselves assumed that some other, yet-to-be-discovered mechanism was controlling the rate of aging. However, in 1993, Cynthia Kenyon and her colleagues at the University of California at San Francisco provided evidence for a genetic influence on aging that wasn't due to reduced fertility. They demonstrated that mutations in a different gene called DAF2 cause worms to live more than twice as long as normal worms. So these old mutant worms actually behaved a lot like young worms. For example, they moved around just as much as the young worms did. And critically, they were still very fertile. Furthermore, this research team was able to show that destroying the reproductive cells in normal worms, and therefore eliminating their fertility, did not actually increase their lifespan. So these findings suggested that the rate of aging itself was under genetic control, independent of fertility. And inspired by these results, in invertebrates, many scientists started looking at similar genes in humans that might be related to healthy aging. And although the jury's still out, there are definitely some promising early results. For example, in 2008, Dr. J. David Kerb and his colleagues at the University of Hawaii looked at the form of 5 hypothesized longevity genes in a couple of hundred Japanese men who were at least 95 years old, a relatively rare population. And they also looked at the form of the same genes in a few hundred other subjects whose average age was 77, a population common enough to act as a control group. Well, sure enough, the very old men tended to have a different form of one of these genes compared with the control group. And since then, this same gene, called the FOXO3 gene, has been linked to human longevity in at least 6 other human cohorts around the world. A final piece of evidence for the hypothesis that some aspects of aging really are genetically programmed comes from the study of patients with genetic disorders. So in very rare cases, genetic anomalies can lead to what looks like extremely rapid aging. For example, patients with Hutchinson-Gilford progeria appear to age at something like 7 times the normal rate. Although they often appear normal at birth, within the first few years of life, they typically lose their hair, their skin gets wrinkled, and they develop many other symptoms associated with old age. Poor eyesight, cardiovascular disease, muscular degeneration, stiff joints, hip dislocations. Unfortunately, this rapid deterioration continues, and so patients with this syndrome don't usually live past their late teens or early 20s. Okay, so now we have evidence from twin studies, from studies in roundworms, from studies identifying genetic differences in very old people, and from patients with genetic diseases. And all these studies point to the same conclusion. Some aspects of aging really are genetic. But why? I mean, why would we be genetically programmed to age and ultimately die? I mean, aren't our genes supposed to be helping us to survive? That's the second major question I want to address. Well, back in the late 1800s, a famous German evolutionary biologist named August Weismann proposed an answer that is often referred to as the programmed death theory. According to Weismann, although aging is obviously not good for the individual, it is good for the species as a whole. In particular, if individuals in a species didn't age and die, then they would compete for food and other natural resources with the younger generations. Therefore, aging and death are actually necessary for a species to thrive. But a very significant problem for Weismann's theory is that animals in the wild don't usually die of so-called natural causes. They usually die from predation, from disease, from lack of food, or other external factors. For example, 90% of mice in the wild die before they turn 10 months old, while mice in captivity live for 2 years or more on average. Likewise, chimpanzees typically live less than 15 years in the wild, whereas in captivity, their median lifespan is more than 25 years. And the same is true for many other species. The bottom line is it doesn't seem like there's any need for a genetic time limit on lifespan because nature herself is dangerous enough that most individuals won't die of old age anyway. So most theorists, including Weismann himself, therefore actually abandoned the programmed death theory of aging. Now, recognizing this key problem, the British biologist Sir Peter Medawar proposed an alternative theory called the mutation accumulation theory of aging in 1952. The basic idea is that natural selection has less and less of an effect as we get older. And that's why genetic variants that lead to deterioration and death in old age don't get weeded out. So, why would the impact of natural selection get smaller as we get older? 2 reasons. First, given that nature is dangerous and that individual animals are getting killed off all the time, there simply won't be that many old animals around. And so if a genetic variant exists that causes old animals to become frail and die, it won't make much difference because the animal isn't likely to reach that age anyway. The second reason is that genetic variants that have harmful effects late in life will have already been passed on to offspring. They therefore won't be selected against as strongly as variants that have harmful effects before reproduction. Now, to illustrate this point, Medawar used the example of Huntington's disease. As you may know, Huntington's is a degenerative disorder that leads to impaired muscle coordination, mental decline, and ultimately death. This is the disease that killed the famous folk singer Woody Guthrie. And it's caused by a mutation in a single gene. So you might wonder why that mutation wouldn't have been eliminated by natural selection. Well, the age of onset for Huntington's disease is typically after age 35. And so many patients have already passed on the mutant gene to their offspring before they even knew that they had the disease. So the harmful genetic variant has its effect too late in life to be consistently selected against. Well, that same argument can be applied to all aspects of age-related decline, and even death itself. Genetic variants that lead to deterioration and death simply won't get selected against if their effect isn't felt until old age. And as a result, they tend to accumulate causing the kind of deterioration that we associate with aging. That's the mutation accumulation theory. Now, Medawar theory has been very influential, but it does have at least one significant problem. Many of the harmful effects of aging begin relatively early in life, often before reproduction. For example, physical abilities tend to start declining around age 20. Consider the world record in the 100-meter dash as a function of age, for example. Usain Bolt was 23 in 2009, the year he set a world record of 9.58 seconds in the men's 100-meter dash. As of 2015, the record for a 35-year-old was 9.96 seconds. For a 40-year-old, it was 10.29. And for a 45-year-old, the record was 10.72. And if we look at the records as we continue to add 5 years, the numbers keep going up. Bottom line, we see harmful effects of aging starting in the 20s. Well, that seems inconsistent with Medawar's hypothesis. In particular, mutation accumulation would predict that harmful effects of age that appear during youth, when reproductive capacity is still high, should be selected against and eliminated. But apparently, they aren't. So why not? Well, in 1957, George Williams at Michigan State University proposed a variant of Medawar's theory that addresses this question. His theory has come to be known as antagonistic pleiotropy. So pleiotropy refers to the well-established fact that a single gene can have different effects within the same organism. And Williams proposed that sometimes these effects could be antagonistic. That is, one effect of the gene might be beneficial to the organism, while another effect is harmful. Now, Williams argued that age-related impairments could be seen as examples of this kind of antagonistic pleiotropy. Specifically, he suggested that a genetic variant that's beneficial during youth might actually be harmful during old age. And following Medawar, Williams assumed that the effects of natural selection would get smaller and smaller as we get older. Consequently, a genetic variant that leads to deterioration in old age could still be selected for if it increases fitness and reproductive success during youth. And for this reason, antagonistic pleiotropy is sometimes called the pay-it-later theory of aging. Now, one example that's sometimes used to illustrate this idea is high testosterone levels in men. So genetic variants that produce higher testosterone levels tend to increase fertility and reproductive success. But higher testosterone levels also increase the risk of developing prostate cancer late in life. Nevertheless, from the viewpoint of natural selection, the benefit of improved reproductive success early in life outweighs the cost of increased cancer risk later in life. And so, high testosterone levels continue to be selected for. That's antagonistic pleiotropy. Now, one specific mechanism by which antagonistic pleiotropy could explain aging was proposed by Dr. Tom Kirkwood at the National Institute for Biological Standards and Control in London. He argued that our body is constantly deteriorating at a molecular level, and it must be repaired. However, making those repairs requires energy that could be used for other functions, like reproduction. So there's an inherent trade-off or antagonism between energy invested in reproduction and energy invested in maintenance and repair of the body. And consistent with the idea that natural selection cares more about reproduction than it does about living for a long time, maintaining the body gets sacrificed. This view is often called the disposable soma theory of aging, because it assumes that the main point of the body, or soma, is to reproduce. And after it's done so, the soma becomes, in some sense, disposable. Okay, mutation accumulation, antagonistic pleiotropy, and disposable soma theory are all attempts to explain why we age, and why natural selection hasn't already eliminated age-related decline and death. But one question these theories don't address is how our genes control aging. In particular, what are some of the actual biological mechanisms that make us grow old and die? That's the question I want to address in the final part of this lecture. And the story starts back in 1961 at the Wistar Institute in Philadelphia. A young microbiologist named Leonard Hayflick was trying to figure out whether cancer could spread from cancer cells to normal cells, kind of like a virus. So his plan was to expose the normal human cells to extracts from cancer cells and see if the normal cells exhibited cancer-like changes. So Hayflick had to grow a bunch of human cells for his experiment. But that's when he ran into a problem. The cells seemed to stop growing after a while. Now, at first, Hayflick assumed that he had screwed up. After all, the accepted wisdom in the field at the time was that if you take care of cells in culture, they'll continue to replicate indefinitely. So Hayflick figured he must have made some kind of mistake, maybe in cleaning the glassware or preparing the culture or something like that. But despite his best efforts, Hayflick's cells always eventually stopped dividing. Well, finally, Hayflick and his colleague Paul Moorhead started to investigate the limits on cell division systematically. In one particularly telling experiment, they put older cells that had already divided a lot with young cells that had only divided a few times into the same culture. And they found that the older cells stopped dividing long before the young cells did. Now, these results demonstrated that the limit on the number of cell divisions wasn't a result of a technical flaw. After all, if the culture was bad or the glassware was dirty, then all the cells in the mixed culture should have stopped dividing. The fact that only the old cells did demonstrated that the results were due to something about the cells themselves. So what is it about the cells that limited their lifespan? Well, one possibility is simply the passage of time. Maybe the longer a cell has been alive, the less capable it is of cell division and growth. But another possibility is that the limitation is based on the number of previous cell divisions, independent of time. To try to distinguish these alternatives, Hayflick froze some of the cells, which kept them alive but prevented them from dividing. He then unfroze them and observed how much longer they could live and divide. And he found that once the cells were unfrozen, they took up right where they left off, and they divided just as much as cells that had not been frozen. So these findings suggested that the fundamental constraint might be on the total number of cell divisions or replications, and is probably independent of time. In fact, it's now well established that most human cells are only capable of about 50 to 70 divisions before they enter what's often called cellular senescence, and they can no longer divide. And this specific limit is now referred to as the Hayflick limit, in honor of its discoverer. So, why is there an inherent limit on the number of times that a cell can divide? Well, about 10 years after Hayflick's original experiments, a Russian biologist named Alexei Alovnikov proposed a theoretical possibility that was subsequently shown to be correct. Alovnikov says that the theory came to him in a Moscow subway station when he thought about DNA replication as being analogous to a train going through a subway tunnel. You see, whenever a cell divides, both new cells obviously need a copy of the genetic material. And so somehow the DNA needs to be replicated. The enzymes that create new DNA molecules are called DNA polymerases. And they work by stringing together subunits of DNA, which are called nucleotides. They string them together one at a time in a single direction. So imagine a train slowly crawling down a railroad track, in which each piece of track is actually a different color. Now imagine a person hanging out of a window in the caboose who's trying to lay down a new track right next to the old one that has the exact same sequence of colors. Well, the train track is like the DNA that needs to be copied, and the passenger in the train is like the DNA polymerase that's copying the DNA. That was the analogy that Alovnikov was thinking about in that Moscow station. Then he thought about what would happen if the train went to a closed-off end of the tunnel. and he realized that the passenger would not be able to copy the very end of the track because the train itself would obstruct his view. Alovnikov proposed that the same thing might be happening during DNA replication. If the DNA polymerase can't copy the DNA all the way to the very end, then the DNA would actually get shorter with every single replication. And he suggested that once the DNA gets too short, then the cell would enter cellular senescence and would no longer be able to divide. Alovnikov's theory thus provided a beautiful mechanistic explanation for the Hayflick limit. But it also raised an interesting question. Why does it take around 50 replications before problems start to arise? I mean, it seems like this process of DNA shortening would lead to the loss of genes near the end of the chromosome, which could potentially cause serious problems much sooner. Well, it turns out that there's a disposable protective cap at both ends of every DNA molecule, kind of like the plastic tips on the end of shoelaces that prevent them from fraying. These caps are called telomeres, and they don't contain any genes. They just consist of the same 6 nucleotides repeated thousands of times. And just as Alovnikov predicted, these telomeres get shorter with every cell division because the very end doesn't get copied. But the telomeres protect the genes themselves, and they prevent them from getting truncated. Now, of course, with enough divisions, the telomeres get completely used up, and they can no longer protect the genes. That's when cells reach the Hayflick limit and enter cellular senescence and stop dividing. However, it turns out that some cells aren't subject to the Hayflick limit. For example, cancer cells can divide indefinitely and are often considered to be biologically immortal. In fact, cancer cells that were removed from a patient named Henrietta Lacks in 1951 have been cultivated ever since, and they're used in countless scientific studies to this day. Likewise, animals that are thought to be biologically immortal, like the lobster and the hydra, they don't show any signs of cellular senescence, despite hundreds of cellular divisions. So what's going on in these seemingly immortal cells that allows them to bypass the Hayflick limit? Well, on Christmas Day in 1984, Dr. Elizabeth Blackburn and her graduate student, Carol Greider, discovered an enzyme that can actually rebuild telomeres after they've been shortened. They named this enzyme telomerase, and it has since been found to be active in about 90% of all human tumors and in all lobster organs. Furthermore, human cells that were genetically engineered to express telomerase were found to continue dividing long past the normal Hayflick limit. Well, as you might imagine, these findings led to a lot of excitement among researchers and to a huge number of studies investigating the extent to which telomerase could slow or even reverse aging. One of the most exciting, but also controversial, was published in 2010 by Ronald DePinho and his colleagues at Harvard Medical School. What they did was to create genetically engineered mice that had abnormally short telomeres and no telomerase activity. However, these same mice would activate telomerase whenever they ate a special chemical. And this gave the researchers experimental control over telomerase activity. At first, they let the mice grow up without feeding them the chemical. And so their abnormally short telomeres were exhausted faster than normal. Sure enough, they found that the mutant mice also aged much faster than normal. But then, they fed them the chemical, which activated telomerase and rebuilt their telomeres. Believe it or not, doing so actually made these mice look young again. Organs that had deteriorated, including the spleen and intestines, suddenly recuperated. The animals' brains were larger, and their sense of smell recovered. Even shriveled testes grew back, and the mice became significantly more fertile. Dr. DePinho described the results as a near Ponce de Leon effect, referring to the explorer associated with the search for the fountain of youth. Now, although these results are obviously very exciting, there are at least two important qualifications to keep in mind. First, the study was not conducted in normally aging mice, but rather in mice that were significantly genetically altered. So the extent to which the results apply to normal aging in mice, much less normal aging in humans, is a very important open question. Second, there's a very strong connection between telomerase activity and cancer. In fact, many scientists believe that telomere shortening is actually an adaptive mechanism whose main role is to prevent cancer. Remember, cancer cells divide without limit, and so a mechanism that prevents cells from dividing indefinitely may actually prevent the development of cancer. And of course, that's exactly what telomere shortening does. So the obvious concern is that telomerase-based therapies might actually increase the risk of cancer. Nevertheless, it's an exciting direction for future research. Okay, let's wrap up. So we've reviewed a lot of evidence that our genes really do influence how long we live. And we've also explored some reasons why, from mutation accumulation theory to antagonistic pleiotropy. And finally, we even learned a little bit about some of the underlying genetic mechanisms, like cellular senescence and telomere shortening. But aging isn't all genetic. As we discussed, twin studies suggest that roughly 25% of the variance in lifespan is due to genetic factors. But that leaves a whopping 75% that isn't. And that raises the possibility that we might be able to do something about aging in general, and about our aging brains in particular. And most scientists definitely believe that we will eventually be able to slow down and potentially even reverse many of the effects of aging. In fact, some scientists go so far as to claim that age-related deterioration is actually a disease, and they view their mission as trying to find a cure. Is there any merit to that idea? And if so, what are the pathological mechanisms at work that lead to aging in the brain and elsewhere in the body? And what can we do about them? Those are some of the questions that we'll discuss in our next lecture.